scientists for their contributions over the past 3 years. Our 5 awardees are really outstanding, and the scope of work really, I think, is fantastically representative of the school. So I just want to thank you for your work, and thank you for the results of your work. Appreciate you incredibly as representatives of our institution. And I'd like to bring up Mary Dickinson, our Senior Vice President for Research.
Okay, I have the pleasure of introducing, thank you.
Yay.
Thank you. Cue the applause. Thank you.
We've got the standing room only, the crowd is moving in, so. We get crowd control in the back.
I have the pleasure of introducing the awardees as Dr. Klotman gives them their medals. So I'm very pleased to do that today. Our first awardee is Dr. Ben Dineen. He is a professor and the Dr. Russell J. and Marion K. Blattner Chair in Neurosurgery and part of the Center for Stem Cells and Regenerative Medicine. Dr. Dineen's lab focuses on developmental gliogenesis, glial control of brain circuit function, and glial-based diseases of the central nervous system, including malignant glioma and white matter disorders. He is working to explain the distinct functions of glial cells in different brain regions, and his work also is tackling the diversity in astrocytes and how they respond to brain injury and how they participate in learning and memory. Over the past 3 years, his research efforts in these areas have produced 12 senior author research articles in high-impact journals, including Nature, Neuron, JCI, Cancer Discovery, and Nature Neuroscience, among others. Dr. Dineen?
You give your talk later.
Yeah, you give your talk after. We'll introduce everybody, give them the medals, take pictures, and then.
All right, so it's right here. Here we go.
You're the trend center here.
Perfect. Let me check. Got it.
All right, thank you. Oh, do you want it?
I'm keeping it.
Okay.
You can take it when you're, we'll leave them all up here.
We want to make sure you
thought
stay
you'd
till
get
the end.
No, this is kind of like the Wimbledon trophy. You get your name on it, we take it.
I'm sure the Nobel Committee has a much smoother process, but okay. All right, our next awardee is Dr. Hannah Al-Sali. Dr. Al-Sali is a professor of molecular virology and microbiology and of medicine in the infectious diseases division. She, since 2016, has served as the PI at Baylor's National Institutes of Health funded Vaccine and Treatment Evaluation Unit here, where she and her team conduct translational research of vaccine candidates, including vaccines for influenza, neglected tropical diseases, and agents that could be used as bioweapons. She is an international leader in translational research of vaccines. I didn't write this. And her leadership and scientific rigor has been recognized with appointments to the World Health Organization, the National Institutes of Health, the Food and Drug Administration, and the Wellcome Trust scientific advisory panels and boards. Dr. Al-Sali played a pivotal role during the pandemic, leading our Moderna vaccine trials, which resulted in 2 beautiful New England Journal papers, and had also led the adaptive trial for remdesivir and the combination trial for baricitinib. Remdesivir is currently the only antiviral medication approved for treating SARS-CoV-2. In addition to her work during the pandemic, Dr. Al-Sali has led clinical trials on influenza pandemic preparedness, the universal influenza vaccine, Zika virus infection, a word I can't pronounce, and another word that I can't pronounce. There you go.
Chikungunya.
Dr. Al-Sali, join us.
It's a main course in some restaurants. I'd like the chikungunya, please.
Other chicken. It's a little sauce on the side.
There you go.
I know she's doing her job. That's good.
I'm married to one of those, by the way.
Okay, the next awardee is Dr. Fasia Kanwal. She is professor of medicine and section chief in gastroenterology and hepatology and a member of the iQuest Center and the Dan L. Duncan Comprehensive Cancer Center. Her research focuses on clinical management and outcomes for patients with liver diseases, including hepatitis, non-alcoholic fatty liver disease, and hepatocellular carcinoma. Her work has provided critical insight into the magnitude of quality problems in liver disease and healthcare delivery, and her high-impact work has led to changes in the Centers for Medicare and Medicaid pay-for-performance programs and set the stage for the first large-scale quality improvement collaborative in cirrhosis. The American Gastroenterological Society has incorporated her findings into clinical practice guidelines for screening and surveillance for hepatocellular carcinoma. Welcome.
You need some help with the pronunciations?
Welcome. It's giving me a tough time today.
Congratulations.
Here you go.
Good.
Thanks.
Great. Next,
I'm keeping them all, so don't worry. It's not gonna be like-
we reuse them every year. No, I'm just kidding.
Yeah, every year we reuse them, recycle, melt them down.
Next, we would like to recognize Noa Lee, Assistant Professor of Neuroscience and a McNair Scholar. Dr. Noa Lee's research aim is in understanding fundamental principles of how brain-wide circuits encode and maintain information and how interactions across multiple brain regions allow the brain to prepare and initiate voluntary movements. Dr. Lee has developed a novel high-throughput approach to studying and manipulating brain function during behaviors, and this approach allows dozens of mice to be studied and tested for voluntary decision-making and enables his lab to connect specific behaviors to molecular properties and activities in identified neurons and neural circuits. Welcome, Dr. Lee.
Congratulations, congratulations. Good, okay. And last but not least.
And last, here comes the sun. Our final DeBakey Research Award recipient for 2021 is Dr. Zheng Shan, Associate Professor of Molecular and Cellular Biology and Medicine, Endocrinology, Diabetes, and Metabolism Division. His research focuses on epigenomic regulatory mechanisms in the context of diabetes, autism, and Alzheimer's disease. He studies how energy metabolism and neurocognition are regulated by diet, exercise, hormones, and the circadian clock. He's made seminal discoveries, including a connection between the central circadian clock dysfunction and the early morning high blood sugar in type 2 diabetes patients, a prevalent condition known as the dawn phenomenon. Dun, dun, dun. Welcome, Dr. Sun.
It's my clock, Gene, that's a problem.
Here's your clock, Gene. We should clean your clock.
Thank you, okay, excellent. So
Okay.
You can have them come up.
Okay, now we'll start the symposium. If Dr. Deneen could join us. And I think your talk is one of these. Can we bring up his slides? Let me see, which one is yours?
That's the one.
I knew it would be the one with the picture. I got it, okay, here we go. Okay. There we go. All right, we're good to go.
Okay, let me test this out here. You guys can see the mouse, right? Great, okay. Well, first off, thank you so much for this award. It's a really great honor to be here with all the prestigious awardees. This is the second time I've won the award, and ironically, I'll be talking about really the same gene that I won the first award with, but in a very, very different context. So as mentioned before, my lab studies glial control of brain circuits and brain tumors. And this picture really sort of illustrates where my lab has gone over the last 5 or 6 years. This big cotton candy looking cell that's pink and green, these are astrocytes. They play a central role in CNS function. In fact, their contributions to neural circuit function has only recently been appreciated. In fact, 15, 20 years ago, it was a little bit controversial as to whether astrocytes or glial cells, because they're not electrically active and they have sort of these passive physiological properties, whether they actually participate in brain circuit function. And over the last 10 or 15 years, it's sort of been realized that they are active players in brain circuits and in brain function. And what I will talk about today is some of their contributions to learning and memory. And the other part of my talk will actually take mechanisms that astrocytes utilize in normal development and describe how those mechanisms are utilized to engender and drive brain tumor formation and brain hyperactivity associated with brain tumors. Okay, so these astrocytes are really important cells. What's interesting is that glia outnumber neurons in the brain. In flies, there's 4 neurons for every glia. In mice, there's 4 glia for every neuron. So as you go through evolution, there's an increasing number of glial cells. They play diverse physiological roles. So there's 3 main types of glia. There's oligodendrocytes, there's astrocytes, and there's microglia, which are immune populations. Astrocytes are a little bit enigmatic functionally because they do so much. They play key roles in regulating neurotransmission and synaptic activity. They play a key role in developmentally in making synapses and in maintaining synaptic structures. They play a key role in providing metabolic support, the blood-brain barrier, so on and so forth. They have at least 10 or 15 different roles that they execute in the normal functioning brain. And because they have such diverse roles, you can imagine that they play key roles in disease. In fact, every single neurological disease has an aberrant astrocyte or a reactive astrocyte component to it. So because they're so important in normal functioning of the brain, when things go wrong in the brain, you can imagine astrocytes are going to be there responding and playing active roles. In fact, one of the publications that this award is for, actually, I'm not going to talk about, actually describes roles for reactive astrocytes from stem cell niches after stroke responses. So these are important cells. How do we get here? So when I started my lab almost 13 years ago, and Peggy, I can see you sort of like, oh my God, 13 years. It's been a while. So the formative studies in my lab were really investigating these 2 transcription factors called NF1A and SOX9 and the role that these 2 factors play in the developing spinal cord during gliogenesis. In the long story short, what we found is that NF1A and SOX9 collaborate to drive developmental gliogenesis. NF1A promotes astrocyte development, blocks oligodendrocyte development, and there's this really cool tripartite chromatin structure that regulates NF1A induction, and all this biology is relevant for glioma tumor genesis. However, some of our studies about 4 or 5 years ago on adult astrocytes really pointed to NF1A and SOX9 in mature astrocytes. So we had studied these things in development, and we were really evolving towards understanding their prospective roles in mature astrocytes in the adult brain. So that leads to sort of the first paper that this award is for, and that is astrocyte control of brain circuits. I don't think that there's a citation here. Yeah, I changed it later. Anyway, so basically what we have here is this is an astrocyte reporter mouse. The green cells basically labels all astrocytes in the adult brain. And the first part of my talk is about what is the role of NF1A in these adult astrocytes in the adult brain, and does it contribute to circuit function in the brain? So this is work done by a postdoc in my lab, Jung-Sung Woo, and a former student, Anna Huang, who's now at UCLA. So to conduct these studies, we needed to bypass the developmental requirement of NF1A. So this required the use of an astrocyte-specific temporal inducible Cree line. So this line is called ALDH Cree ER. It allows us to delete NF1A specifically in astrocytes in the adult. So we made a series of mouse lines. I'm embarrassed. There's probably 12 different lines that have NF1A. It's really kind of ridiculous, but we made all these lines, and the idea was to delete NF1A after development in these sort of early adult stages and ask what happens to astrocytes and to mice when astrocytes lack NF1A. So here you can see we have really nice expression of NF1A in these adult astrocytes in roughly 4-month-old mice. When we add tamoxifen, we wait roughly 2 months. We can see we have very efficient deletion of NF1A in astrocytes, and you'll note in the brainstem there is some expression of NF1A in some neuronal populations, and that expression is retained. So this is specific deletion of NF1A in astrocytes in the adult brain. So what happens to astrocytes when we knock it out? So what we noticed immediately was that things were actually okay. The mice were fine, they were moving. I thought for sure the brain was going to melt and the mice were going to die, but they didn't. So we looked carefully. My student looked very, very carefully. She found something very striking. She found that if you look in the knockout, she noticed that astrocytes in the hippocampus look very different. Normally, astrocytes have this very bushy, ramified morphology. You knock out NF1A in the hippocampus, they're stunted, they look sick, they're very they lose this elaborate morphology. Brainstem, they seem to be unaffected. Olfactory bulb is unaffected. Cortex is mildly affected, and that's sort of quantified here. So what this tells us is that there's a region-specific dependency for NF1A expression to maintain astrocyte morphology, specifically in the hippocampus. So that raises the question, does this translate to region-specific target genes? So because it's a transcription factor, we wanted to know what are some of its target genes. I won't go through all the gory details. There's a lot of RNA sequencing, a lot of chip sequencing. But the long and the short of it is that NF1A is able to bind DNA very efficiently in the hippocampus, but strikingly, it doesn't bind DNA in the olfactory bulb. So when you remove it from the olfactory bulb, it doesn't really matter because it's not binding DNA or doing anything. But in the hippocampus, it binds DNA, and when you remove it, things get a little bit weird. So next we look at some of the target genes, and the target genes made sense, right? You have sort of integrin binding, actin filament, collagen binding. All of these things are important for maintaining cell morphology and this elaborate morphology that astrocytes exhibit. So this was sort of one of the mechanisms that we found. So this raises the next question is, because the hippocampal astrocytes have this messed up or stunted morphology, what happens to astrocyte function and circuit function in the hippocampus? So the hippocampus, to non-neuroscientists, because I'm not a neuroscientist, I'm learning this on the fly, is basically a great place for studying circuits. It's a really well-defined trisynaptic circuit that is really responsible for learning and memory, and astrocytes have previously been implicated in regulating sort of circuit outputs in the hippocampus. The other thing that we need to know is that all roads into and out of an astrocyte are mediated through calcium activity. Astrocytes suck up neurotransmitter that causes rises in calcium, and there's calcium-dependent release of gliotransmitters, which is a little bit controversial, but they do release things that neuroactive compounds that influence circuit function. So we wanted to know what happens to these activities in these NF1A deficient mice. So can we quantify calcium? So we use this optical sensor called GCamp. So we put this optical sensor called GCamp into the astrocytes in the mutant and the controls, and we looked at GCamp activities, and we found that there's a huge, dramatic reduction in calcium activity in these astrocytes, which is not surprising given how sort of stunted and messed up they are. Next, we looked at physiological responses. So long-term potentiation is the physiological readout of synaptic plasticity and learning to memory. And it's thought that that measurements of LTP are correlated with learning and memory functions. And so it's been shown in other studies pharmacologically that astrocytes can influence LTP responses in neurons. So what happens in our mutants? So we basically stimulated the Schaefer collateral. Again, I learned all this over the last couple of years. It's like really cool stuff. And what we found is that in the knockout, you basically have no LTP. So you have this stunted morphology, you have reduced calcium activity, and you have the neurons aren't behaving properly. So the final question that we asked, and this is probably the most important one of all, is what about behavior, right? Because behavior is the output of this circuit function. So we did a series of behaviors, not just learning and memory, and strikingly, the only behaviors that were affected in our mouse were learning and memory, which makes sense because the hippocampus controls learning and memory, and that was the only region that was really impacted by loss of NF1A. So we did contextual fear conditioning. You can see there is reduced fear conditioning in the knockouts. Next, we looked at a novel object and also spatial working memory, and you can see that there are decreases in each of these behaviors in the mice lacking astrocytic NF1A. So collectively, what this study tells us is that basically there's this region-specific dependency. In the olfactory bulb, NF1A doesn't bind DNA, doesn't really do much. In the hippocampus, it binds DNA. When you remove it, the astrocytes get stunted, and you have defective circuit function in these mice. And this was featured on the cover of Neuron last June. This is like really cool because it's astrocytes in its space, right? You get the whole connect. And we're in Houston, right? It all kind of fits together. Okay, so that's sort of the first part of my talk. The second part of my talk really is completely different, but yet related. So the basis of this project is really the functional genomics of cancer. And it's known that, over the last 20 years, we spent millions and millions of dollars sequencing cancer genomes. And we've now annotated in the COSMIC database over 3 million different cancer mutations. However, We only know what 5,000 of those mutations do. So if we're ever to achieve the dream of personalized medicine, we need to understand what these mutation cohorts are doing. And over the next 20, 30 years, I believe in others that functional genomics is going to be really key, making sense of all of these mutations and all these variants. So this is sort of where this project started, the main question. But it took us somewhere completely crazy. So to do this, this is all based upon this in utero electroporation-based model of glioma that my lab established about 6 or 7 years ago now. And the idea is that we can inject the embryonic cortex with piggyback driving oncogenes or guide RNAs and Cas9 to delete genes. We can introduce these constructs into the embryonic cortex. We can stuff the embryos back in the mom, they're born, and we can get tumor-bearing mice. The nice thing about this system is that pregnant mice is relatively cheap, has an intact immune system. These mutations are introduced in cell of origin tissues and in an autogenous setting, and it is relatively robust. A single pregnancy can give us 6 to 10 tumor-bearing mice. And this is what the tumors look like. So basically, you overexpress RAS, which is not a great mutation for glioma, but you get really nice tumors. I'll draw your attention to this triple CRISPR, where we delete NF1, P10, and P53, 3 key tumor suppressors deleted in glioma. And by postnatal day 70, we get these robust gliomas. We can fax them, we can image them, and we published a series of papers using this model. So this is what led to this functional genomics, this interest in functional genomics. And so what led to it was really sort of playing with the CRISPR combinations, the guide RNA combinations. So here we have the triple CRISPR, where we have this sort of median survival is roughly 100 days. If we remove loss of P10, that's extended to roughly 220 days. So this gives us a nice dynamic range. Same thing with removing loss of NF1, we get an extension of survival. So the idea was, could we use this dynamic range as a platform for screening for new driver variants? And so what's interesting is that the PIK3CA mutations are pretty common in cancer. Gain of function PIK3CA mutations function or phenocopy a loss of P10. So the idea would be, could we screen a cohort of PIK3CA mutations and ask whether they restore this double loss back to the triple CRISPR survival. The same thing with EGFR. We do the exact same thing with EGFR. So loss of NF1 functions through MAP kinases. EGFR functions through MAP kinases. Do activating EGFR mutations rescue a loss of NF1? I won't go through the EGFR stuff, but I will focus on the PIK3CA because that's the most interesting. So can we functionally screen these in the system? And we can. So we took all 27 variants that are present in glioma, and we basically put them with this double CRISPR combination. We electroporated this cocktail into the embryonic cortex. When tumors formed, we sequenced for barcodes. I left out that each variant has a barcode, and it allows us to identify it. So, let me go back. Oh, that's fine. So basically, this is the result of the barcoded screen. You can see here, R88Q, C420R, 545K, and 1047. These are all the variants with barcodes that were enriched in the tumor. So these were thought to be the leading candidate drivers amongst this cohort. But this is just a screen. We went back and individually validated. Yes, each of these lines is over 40 mice. So we went back and individually validated each of these variants in the context of a double CRISPR. And just for reference, the gray line here is the double CRISPR. The black line is the triple CRISPR. So that's our dynamic range. Each of these identified drivers is very, very strong in this context. The blue and the red lines are, this is C420, and the red line, I believe, is 545K. So you get a sense of how strong these are in this context. So we had this result, and I was not impressed. As Peggy knows, nothing really impresses me. And so we wanted to know more about what this meant, right? So we sequenced, like any modern molecular biologist, we sequenced the tumors. So these are PIK3CA variant-driven tumor transcriptomes on a PCA plot. You can see you sort of have this core set. However, C420R and H1047R are very different from one another and very different from the core set. So this was very exciting because we had tumors that were driven by variants that differ by a single amino acid, and they generated transcriptomes that were vastly different. This is a very striking find. This is all done in vivo, too. This is very striking. So we then asked, well, what separates them, right? That's a natural question. And of course, proliferative genes came up, but also there is this synaptic set of genes. And what does synapses have to do with cancer? I see Paul, he's perplexed. I was too. So what does synapses have to do with cancer? So we wondered, like, what does this have to do? Is this important? So what's the significance of the synaptic gene set? And so we know that, if you have a brain tumor, you often present with a seizure. Roughly 50% of patients that have a GBM will present with some form of seizure, some form of hyperactivity. And you know that microcircuit alterations, an excess in excitation, a loss of inhibition can result in seizures. So we wondered, is there something about these variants in these tumors and seizure onset? So we tested it. So what we did was we generated these tumors in this model where we overexpressed individual variants, and we did EEGs. And you can see here, R88Q, representative of the core cohort, really didn't have any seizures until much later. C420R, H1047R were active very, very early. They induced seizures very early onset, very surprising. So next we looked at synapses, and hold on, oh, this takes a while. Uh-oh. We looked at synapses, and when we stained for, there we go, okay, good. So basically, we stained for synaptic markers at the peritumoral margin outside of the tumor, and what we found was that there was an increase in synapse formation outside the tumor. There was an increase in excitatory synapses and slightly less inhibitory synapses, telling us that the tumor is communicating with neurons at the margin to drive this hyperactivity. So what does this? I'll skip quickly over this next slide, if it gets there. Okay. Basically, we did some in vitro experiments, points to a non-autonomous mechanism. It's known that astrocytes release factors that promote synapse formation. We wondered, and there's two families, glypicans and transpondins, that do this. Turns out members of the glypican family are enriched in C420R tumors, which are very strong drivers of this hyperactivity. So we investigated glypican-3 function, and it basically functions in the same way. When we remove glypican-3 from the C420R tumors, survival is altered, and you don't get the early onset of spiking or hyperactivity. Gain of function on its own also gives you a very early onset of these phenomena. So this is sort of the summary. We have a platform. We have related variants, have distinct transcriptomes. They manipulate neuronal microenvironment, and GPC3 is a lead candidate. And finally, I have 2 slides, Mary. I'll be done. So finally, the functional genomics studies led us to the sort of cancer neuroscience initiative, and we've recently submitted a program project grant on this topic of cancer neuroscience. And we submitted the grant, Mary, 2 days ago, or last week. So fingers are crossed, and we're hoping that we can get this through. I also want to briefly acknowledge my lab. That was me with NF1A long ago, and you can't do any of these things without an excellent team. And this is my lab over the last 12 years. Great team. I don't deserve them. They are awesome, and I'm just a hobo with a gene. And finally, I want to acknowledge my dear friend Ken Scott. Ken Scott is he passed away with cancer over 4 years ago. He was my partner in crime with all these functional genomics studies. He's the one who dragged me kicking and screaming into the genomics field. I'm not a neuroscientist. I'm not a genomics person. I'm just a biologist. But Ken was really the inspiration for these studies. And in fact, we just submitted the U01 that we got with him. We resubmitted it, or renewed it, sorry. So his legacy will live on through that. And finally, I want to acknowledge my funding, particularly the Winterman Foundation, who has provided an enormous amount of support over the years. And I'm sorry for being over, Mary. And I'm done. Thank you.
We're significantly over, so I'm going to hold you back
Yeah, no
on
questions.
It might have been 3 or 4 slides of acknowledgements. I don't know.
Probably was.
You can say no to that, so
Thank you.
thank you so much. Dr. Al-Sali? Let me actually see if I can get your talk up. Okay. They're all titled the same thing, making this super easy. This one, yeah, this one. Okay, there we go. Fantastic. All right, take it away.
Thank you all. Thank you for the committee for choosing me to get this award. I will walk you through some of the data and then the publications that sort of prompted this nomination and this award. Lest you forget COVID, this is based on the vaccine and therapeutics developments during the pandemic. It was December 2019 when we found out about the cluster of pneumonia in China. Fast forward, it is one of the deadliest pandemics recorded with, as of 27th of September, when I had to submit my slides, there were 42 million cases in the US, 684,000 deaths, and now we know it's 700,000. The trends are improving, as we see here, but there's a lot of work that remains to be done in terms of control and management. From the get-go, we were involved in the therapeutics and the vaccine aspect. From the therapeutics aspect, we were involved as a VTU in the adaptive COVID treatment trial, which is a NIAID-funded trial that was one of the first and largest ones to launch right after COVID. Act 1 evaluated remdesivir as a treatment against placebo. Act 2, baricitinib with remdesivir versus remdesivir alone. Act 3, remdesivir with inhaled interferon versus remdesivir only. And Act 4 was a head-to-head comparison between steroids and baricitinib on a background of remdesivir. Act 1 and Act 2 data are in the public domain, and I will be sharing some of those data. The Act 1 trial, we randomized individuals hospitalized with COVID-19 to receive either remdesivir or placebo in a 1-to-1 ratio. The study was double-blind, placebo-controlled. What we found after randomizing over 1,000 patients, 541 got remdesivir, 521 got placebo, is that the median recovery time decreased from 15 to 10 days. The data analysis was compelling enough for the DSMB to require that we stop and unblind whoever remains on the study so they can go on and getting remdesivir. The study was not powered for mortality. The mortality on the trial was 11.4 on the remdesivir arm versus 15.2 in the placebo group. So the numbers indicate a 27% reduction. That was not significant, but again, it was not. That was not the power for it. When the study was analyzed using the common methodology of having the outcome being the progression to mechanical ventilation or death, then remdesivir did result in an improvement with a hazard ratio of 0.67 for those on remdesivir to reach the outcome of mechanical ventilation or death, and that was statistically significant. Then we moved on to evaluating baricitinib, which is an immunomodulator, inhibitor of the JAK pathway in the management of hospitalized COVID-19 patients. With the knowledge that was easily gained that COVID-19 results in a dysregulated immune response in a fraction of individuals. So the Act 2 was also randomized, double-blind, placebo-controlled, remdesivir with baricitinib versus remdesivir with placebo. And the patient population was the same, those hospitalized with COVID-19 pneumonia. The primary outcome was also time to recovery. The study enrolled 1,033 patients with a mean age of 55. Little over half were on oxygen, 55%, and 21% were mechanically ventilated. In this study, the median time to recovery was 7 days for those on baricitinib versus 8 days for those on placebo. The difference was statistically significant. The other key outcome that was reached was a 30% higher odds of improvement in their clinical status by day 15. Also on this trial, the 28-day mortality was improved, but not statistically significantly so. The trial was not also powered for mortality, but the difference was 5.1 in the Bari group versus 7.8 in the placebo group. Both of these drugs went on to being approved for the treatment of hospitalized COVID in the case of remdesivir, or authorized in the case of baricitinib for hospitalized patients with COVID-19 needing oxygen. Since then, there have been follow-up studies or studies done by others, and we do know now that baricitinib, in a study that was double-blind, also randomized and controlled, placebo-controlled, that baricitinib does offer a mortality benefit that was statistically significant. And in the case of remdesivir, multiple effectiveness analysis did demonstrate a mortality benefits in the real-world scenario. Moving on to the other aspect of the COVID-19 management that we were involved with, which is the vaccine. The study, the COVE study, was a phase 3 clinical trial that evaluated the efficacy of the messenger RNA 1273, which is the Moderna vaccine. Baylor was chosen as the lead site, and I was the national PI with the study, along with Drs. Baden and Dr. Essing. The study enrolled 30,415 healthy individuals who were adults over the age 18 years of age or older. They needed to be in stable health condition. They could have underlying diseases. If they had HIV, it needed to be treated. We excluded immune-compromised or significantly immune-compromised individuals. And as a whole, we enrolled 40% or more who were older than 65 or those with risk factors for severe disease. Individuals were randomized 1-to-1, vaccine or placebo, given 28 days apart. And the primary efficacy analysis was the occurrence of COVID in individuals who had no evidence of previous COVID or SARS-CoV-2 infection, starting at 14 days after the second dose. Here I'm going to spend a minute on the trial population. We see that we had a good representation of men and women. 52% were men, 47% were women. The mean age was 51, with 24% of the study, or close to 25%, being those 65 years of age or older. We had also a good representation of Latinx individuals, with 20% being Latinx. And the race and ethnicity had to be representative of the demographics of the country, and the study actually had to sort of adjust midway in order to stop enrolling Caucasians and enroll those belonging to other ethnic and racial group to ensure a good representation in the study. And we did achieve that eventually. In the trial, only 2.2% were positive for SARS-CoV-2 at baseline, and we did that by testing the antibodies prior to vaccination. And you can see that only 182 had a positive PCR that they didn't know about just on screening. Individuals with risk factors for COVID-19 were also well represented, with those with chronic lung disease, cardiac disease, diabetes, liver disease, and HIV. And what we found is that at 2 months, and these data were published back in December, that the vaccine was 94% efficacious at preventing symptomatic COVID. The patients on the trial were continued to be followed, and no one was unblinded until we received the emergency use authorization, which allowed a median follow-up of 5.5 months. And last week, we published the data on the follow-up up to 5.5 months median follow-up. And we see here that the vaccine efficacy was maintained through 5.5 months at 93.2% for all symptomatic COVID, starting 14 days after dose 2. And when we look at severe COVID-19, as defined on the clinical trial, we found that the vaccine efficacy was at 98.2%. 82%, and that was maintained again for 5.3 months, median. There were different permutations of COVID-19 occurring on the trial. Those that met the definition, and these are the COVID-19 and the severe COVID-19, but there was also what we call a secondary definition, which is having just one symptom, maybe headache, and a positive PCR. So that secondary definition was also at 93% efficacy. Death from COVID, we only had 3, but they were only in the placebo group. COVID-19 after 14 days post first injection, COVID-19 regardless of previous SARS-CoV-2, all the numbers kind of hovered in the same range. When we look at asymptomatic COVID, the vaccine was 63% efficacious at preventing asymptomatic COVID, and that is using either the swab or seroconversion, asymptomatic seroconversion. When we look at the vaccine efficacy by study subgroups, and that's why there was such emphasis on representing the old, those with belonging to different racial and ethnic minorities, those with underlying illnesses, and those with different exposures in terms of healthcare workers, etc. The vaccine also, we can see here the point estimates all hover around the same value. The liver disease, we only had 5 cases in the placebo, 1 in the mRNA, so the small number and hence the wide interval, but certainly in the right direction as well. So, of course, and a lot of these data were replicated in real life. And even with the Delta onset, we had diminution in the efficacy of the vaccines against symptomatic COVID, but not so much against severe disease. So is it the antibodies? What are the correlates of risk or the correlates of protection? So this is the other study that just was placed on medRxiv a few weeks ago. We evaluated the antibody levels at the time points of day 29 when they got their dose 2 or day 57, which is a month after dose 2. And looking at those who had COVID-19 on the study, the breakthrough cases, with 47 starting at after day 57 and 8 occurring in this intercurrent period. Of note, by day 29, 100% of the participants on the trial had converted with antibodies against the spike and the receptor binding domain. The 50% viral neutralization was 82% by day 29, and the 80% pseudoviral neutralization was 64% by day 29. By day 57, there was 100% response for all assays. There was a high correlation between the IgG results and the pseudo-neut results. And here's what we see. In blue are the intercurrent cases. In orange are the cases that occurred post 57 days or 1 month after dose 2. And in purple are individuals who are non-cases. We can see that there is a trend with rising that those who were non-cases tended to have higher antibody levels, and this was statistically significant, compared to those who were a case after day 57, comparing to those who were a case after day 29. But also, there's a lot of overlap. And the same was true on day 57. If we look at the neutralizing titers, also the trend persists in that there's a lot of overlap in the level of antibodies in these 3 groups, but the trend is there is better protection at higher antibody levels. So in conclusion, the vaccine efficacy, increased with the antibody levels, whether it's binding or neutralizing, but there was 50% vaccine efficacy that occurred with no detectable antibody responses. So it's not all the measurable antibodies. And mathematically, when you extrapolate this, and the best thing the biostatistician told me, I could say this, it's a fancy logistic regression. 68% of the vaccine efficacy is mediated by the antibody titers as measured. So there's a lot that is unexplained by what we are measuring. So the next phase is that we'll continue to follow the participant in the phase 3. After the booster declarations in the last month, we had to amend the protocols. So now we are going to vaccinate individuals. They already got the 0-1, but now they're going to be 0-1-8 versus 0-1-13 and look if there's any difference in efficacy there. There's a mix and match study going on to see if we can boost with different products for people primed with any of the EUA vaccines. Pediatric vaccine studies already started. A new generation COVID-19 vaccine with self-replicating RNA and other T-cell based vaccines. And hopefully soon, non-COVID research. I want to thank my colleagues at the VTU, Drs. Whitaker, Healy, Atmar, Keitel, Piedra, Akamine, and Nicholson, the IDCRC leadership, the VTEU staff, some of whom remain with us, some of whom moved on, and the committee and Dr. Giordano. Thank you.
Fantastic talks. Let's see. Okay. All right, Dr. Conwall, very excited.
Thank you. Thank you very much. And just like everyone else, it's really a true honor and a privilege to receive this prestigious award. A very exciting day. I want to actually start by thanking people, my team members, my collaborators, co-investigators, VA, iQuest, Baylor College of Medicine, and Rolando, without which I wouldn't be here today. So thank you very much. I am a physician scientist, gastroenterologist, hepatologist, and a health services researcher, so a little weird combination. I did my health services research training with my GI training and had my faculty position at St. Louis University for the first 5 years before I moved to Baylor. Now, about 10 years ago, so I've been here at Baylor. I serve as a section chief for gastroenterology and hepatology in the Department of Medicine. My research has focused on liver. But as I think about it, it has two broad themes. And the work that I'll present today will be in these two broad themes, mostly focusing on studies that we've done in the last 3 to 5 years. One of the themes is to look for significant associations with different chronic liver conditions, different risk factors, and risk of developing HCC, hepatocellular carcinoma. The overarching goal of this research program is to really improve risk stratification of patients who are at risk for this condition. The second parallel theme is to look for how we deliver care to patients with chronic liver conditions, mostly focusing on a high-risk group of patients with cirrhosis of the liver, where liver is replaced by scar tissue. And the overarching goal of this research is to really figure out a way to improve how we deliver care. So as most of you here would know, cirrhosis is the final common pathway for all chronic liver conditions. And it is also a key underlying lesion for liver cancer, hepatocellular carcinoma. I'll use the term HCC during the rest of my talk. So for all these risk factors, hepatitis C virus infection, chronic infection with hepatitis C virus is the most important, and it is the leading cause of liver cirrhosis and liver cancer in the US. More recently, we're seeing a shift with another condition, non-alcoholic fatty liver disease, or NAFL, as I will refer to it, is emerging as an important risk factor for cirrhosis and for HCC. And our group is the first one to really quantify the magnitude of association between NAFL and liver cancer that I'll share with you in the next few minutes. But before I go there, as I mentioned before, hepatitis C virus infection is and has been the leading cause of HCC in the US, but that was before we had these new direct-acting antiviral agents, or DAAs. These treatments, they are very effective, and they can cure hepatitis C infection in most patients with this condition. But earlier data, when these drugs came out, suggested that they may be associated with HCC risk. They may actually cause people to develop or advance HCC, raising substantial safety concerns. So our group was the first one to really address this question, and we conducted one of the first large-scale studies that looked at over 22,000 patients in the national VA healthcare system who were treated with these drugs. And of these 22,000 patients, 19,000 received cure or sustained virological response. And as we followed this group of patients who were treated with these agents, we found actually that SVR or cure was protective. Patients who had SVR had a lower risk of HCC compared to those who did not have SVR. And this finding was later on replicated by many other studies, really collectively demonstrating that DAs were indeed safe to use. We followed this more because our goal was also to find out among patients who will be cured of this condition, which many of the patients now are cured of the conditions, what are the other risk factors that could still explain residual risk of HCC, because that could guide clinical decision-making. And the most important risk factor was underlying advanced fibrosis, scar tissue, or cirrhosis of the liver. We found that the risk of HCC was high in all patients with cirrhosis, regardless of how old they were, what was their gender, race. It was high all across, and the overall estimate was about 1.8% per year. This number is important because that is above the threshold beyond which HCC screening or surveillance is cost-effective. So this risk was high in all patients with cirrhosis of the liver, and it was low in all patients who did not have cirrhosis by the time they received the treatment, except for a very small subset of patients who had a high FIB4. FIB4 is a non-invasive test. It looks for presence of advanced fibrosis. So the story is that if the patients have progressed to advanced fibrosis or cirrhosis by the time they receive these treatments that can cure their infection, they still remain at a high risk for HCC. So these data were important because they showed that, yes, DAAs, they do have a beneficial effect overall, but there is an important subgroup of individuals, with those with cirrhosis, where the risk remains high enough to require ongoing monitoring and surveillance. And these data informed clinical practice guidelines at that time. More recently, we've followed this cohort over the next 3 to 5 years to really look at whether the risk of HCC or liver cancer drops off during longer-term follow-up. And this is because smaller studies suggest that liver fibrosis can regress with time once infection is cured. If you extend that forward, it is plausible that the risk of HCC could also fall as time from treatment elapses. But when we look at our cohort, that's not the story. The risk remains high enough, at least for the first 3 to 5 years of follow-up since the time of completion of treatment, that this group needs to continue with ongoing monitoring and surveillance, and again, with significant clinical implications. I'm going to shift gears to non-alcoholic fatty liver disease, or NAFLD. This is a hepatic manifestation of metabolic dysfunction. And with the rising prevalence of obesity and diabetes, it is becoming one of the most important causes of liver diseases, with the estimated global prevalence of 25%, so 1 in 4 individuals in the world, they have non-alcoholic fatty liver disease. And it is important because individuals with NAFLD have a high risk of overall mortality, high risk of liver-related mortality, and our group showed they also have a high risk of liver cancer. In this study, we followed and compared patients with NAFLD who were identified based on validated definitions from electronic medical records in the national VA healthcare system with a control group without NAFLD or chronic liver disease. And we found that NAFLD was associated with a substantially high risk of liver cancer. This risk was evident earlier on and became more evident as we followed the patient over time. And overall, the relative risk was 7 compared to those without NAFLD. So again, showing significant association with HCC. And it is problematic, as I mentioned before, because there are going to be many patients with this condition that we will see in the next decades to come. The story continues with cirrhosis being the underlying lesion for HCC in NAFLD as well. Among patients who had NAFLD, the risk of HCC was higher in those with cirrhosis. Again, really pointing towards need for ongoing surveillance and screening in that subpopulation. Collectively, the studies that I showed you have really informed clinical guidelines, more recent ones, for surveillance and screening for both for patients with hepatitis C virus infection that I actually had an opportunity to work on, as well as for patients with NAFLD that Dr. Sarag took a lead on. So in the next half of my presentation, I'm going to shift gears towards the other focus area for our research, which is looking at healthcare delivery, focusing on patients with chronic liver disease, mostly cirrhosis of the liver. These series of studies started with us identifying, developing, and validating quality of care metrics for patients with cirrhosis, followed by identifying data, pooling data together, looking at processes of care delivery, and more recently has shifted towards identification and implementation of interventions to try to improve outcomes of these patients. So I'll share some of that journey with you. Our group was the first one to highlight that there were gaps in how we deliver care to patients with cirrhosis of the liver. So on your left-hand side, these are performance scores across different ascites care metrics when there's fluid collection in the abdomen given because of cirrhosis, and you can see the variation. They varied substantially from as high as 90% getting the right care to as low as 20% getting the right care. And overall, less than a third of patients in this study received all indicated care that they needed for their ascites management. And it is not just that these processes of care have gaps. The outcomes of this patient population of patients with cirrhosis has not really improved much over the last decade. So on your right-hand side, these are graphs from over 100,000 patients who were hospitalized with cirrhosis from 2004 and 2013. And over this one decade, if you look at the dashed line, the in-hospital mortality fell over this time, but this reduction in in-hospital mortality was not associated with any reduction in 30-day mortality if you start following them from the date of admission. So basically, no difference in short-term outcomes in this patient population over the whole decade, which is very different than things that we see in other chronic conditions such as congestive heart failure. So one would say, it's very difficult to improve outcomes for patients with cirrhosis of the liver because the real only curative treatment is liver transplantation. And many patients don't benefit or get liver transplantation because we don't have enough livers. Liver allograft shortage is the main reason why we have low transplantation rates. But we recently also showed that there are other failures that are potentially modifiable in the complex transplantation process that they do impede access to liver transplantation. In this large study that just was recently published, there were 34,000 patients with cirrhosis who otherwise met clinical indications for liver transplantation. Only 4.5% were referred for liver transplantation. 3% were listed for liver transplantation, and 1.8% were eventually transplanted, despite the fact that they had indications for liver transplantation. And as part of this study, we really did a very deep review, abstracted medical records of many patients to try to figure out any reasons that we could identify why a patient that should have been referred did not get referred. And potentially modifiable medical and psychosocial comorbidity explained most of the instances of under-referral. So again, pointing out that, yes, missed referral or delayed referral is a potential barrier to transplantation, but the larger, bigger underlying challenge is to really identify, support, and modify potentially modifiable comorbidity in this patient population. So based on these data, we are developing a program which will identify patients earlier on who have potentially modifiable conditions, try to intervene upon them earlier on to try to enhance access to liver transplantation. This is a grant that's under review right now, got a good score, but if it's funded, we will be pilot testing it in 3 different VA hospitals. But some of this work that I just shared with you has had implications outside of our local context. This work has served as a foundation for American Association for the Study of Liver Diseases, which is our premier liver disease society, their guidance on quality measures around cirrhosis, and I had an honor to lead this as the chair of the committee. And these measures now are being implemented as part of a Cirrhosis Quality Collaborative. Cirrhosis Quality Collaborative, it's the largest quality improvement initiative, again, from AASLD. It is a multi-site learning health network, which is right now being implemented at 10 different sites, including us here, Baylor College of Medicine and Baylor St. Luke's. I am co-PI for this initiative. And it is, I think, where we need to go in terms of really, if you want to work towards translating findings from clinical studies to really clinical practice, where we have a platform, a laboratory, where we can collect the data, act on the data, and improve care, combining quality improvement methods and research. So this collaborative, it is right now developing a centralized web-based registry that is collecting patient-reported outcomes data, combining it with electronic medical record data from all patients who are seen with cirrhosis at 10 different healthcare systems. And combining this, I think, will provide the platform that we need to really improve the care and treatment outcomes of patients with cirrhosis by implementing quality improvement initiatives. So I'm very excited about this. I will wrap up and try to get us back on time. But before that, I wanted to thank all the organizations and funders that have supported this work. Most of the work around risk assessment and risk stratification for HCC is currently funded by NCI, DOD, and CPRIT, and really many thanks to the VA HSR&D, VA Query, and the AASLD for supporting our work around quality measurement and quality improvement. And I'll end by thanking everyone again, my team especially. This award is for all of us. So if you're watching, thank you.
Fantastic. Okay. Dr. Lee. There we go. Fantastic. There you go.
Thank you.
Thank you.
Okay, I want to thank the college and the leadership for this great honor, and also Paul for nominating me for this award. And I want to thank you for this opportunity to share our research with you. Okay, so my lab studies the neural basis of goal-directed volition. Okay, so my lab studies the neural basis of goal-directed volitional movement. So when you're looking at this slide right now, your eyes are currently moving at the rate of 2 to 5 times per second, okay? And these movements your eyes make are highly goal-directed and very precise. To illustrate this, these are the eye movement traces recorded in human subjects when they're looking at this painting. And the human subject was asked to either remember the position of people and object in the room or judge the age of the people. And you can see when looking at the same visual scene, depending on what they're looking for, the eye position takes very different flight paths. In one case, scanning the room broadly, and in the second case, selectively foveating on people's faces. So this example just shows that goal-directed movement is not something that's hardwired in the brain, but rather the brain seems to have this ability to be able to, on the fly, plan the appropriate movement depending on the current goal at hand. So the neural mechanism of such planning is preparatory activity. And it's long known that in motor cortex, neurons become active long before volitional movement. And in humans, this can be measured in EEG. This has been often referred to as readiness potential. And in animal models, such as in non-human primates, this can be directly recorded from the electrical activity of single neurons. So this is the example taken from the original study by Tanji and Everett, in which they used a microelectrode to record from single neurons in monkey motor cortex, and the animals was instructed to either push or pull a mechanical lever. And these ticks show individual action potentials recorded from a single neuron, and you can see this neuron exhibit this barrage of activity shortly after the sensory instruction was given, but long before the animal made the movement. Now, interestingly, this activity predicted what the animal is going to do. So in this case, you can see there is this robust wave of activity before a push movement, but if the animal made a pull movement, this activity was absent. And interestingly, even when the animal made a mistake where it pushed in error, this activity predicted what the animal is going to do next. So this preparatory activity is widely thought to be responsible for volitional movement, and really understanding this activity pattern is key to understanding how the brain is able to flexibly program volitional movement. And understanding this activity will likely open new sites of treatment, interventions for treatment of movement disorders. However, at this point, how this activity pattern is generated in the brain is not really well understood. In practice, probing this activity in human brain or even in non-human primate brain has been challenging. And part of the difficulty is that this activity is widely distributed across many brain areas and over a very large spatial scale, so making it very difficult to track down and manipulate. So for the last several years, my lab has been developing a mouse model to study the neural mechanism of preparatory activity. The mouse brain, the reason for that is the mouse brain is very small, so it is very accessible from experimental manipulations, making it very amenable for mechanistic dissections. And we developed a behavioral paradigm to engage motor planning in the mouse. So we train mouse in a simple decision-making task. The animal receives a sensory instruction. In this case, an object is presented at different locations, and the animal uses their whiskers to measure object location, and they report object location using directional licking movement. So if object is in one position, the animal lick to the right, and if the animal is, if the object is presented in a different location, animal lick to the left. Now, importantly, after we give the animal sensory instruction, there is a delay period before they make the movement, and this provided us a time window to look at brain activity that is driving the upcoming movement. So just to give you an idea, this is a video of a mouse now performing this behavior. You can see the object is being presented briefly, after which there is a brief delay period, and then the animal starts making the goal-directed movement. So this animal is performing this behavior quite well. This is after about 3 weeks of training. And you can see the animal is head fixed here. This is very important. It gives us stable access to the brain for a wide range of recording and manipulation. And a single animal can perform hundreds of these trials in a single session, which give us an opportunity for repeated measurements and perturbations. So in our previous work, we have identified a single region in frontal cortex in the mouse that is critical for motor planning. We call this region anterior lateral motor cortex, and we found that if we silence the activity in this region, specifically during the delay, this will impair the animal's ability to make correct movement subsequently. And interestingly, if we record the electrical activity from this region, we can encounter neurons which show preparatory activity during the delay period that predicts what the animal is going to do next. And so that is illustrated in this example neuron. These individual dots indicates individual action potentials fired by this neuron. And these are activity across several trials of the same behavior that has been aligned to the behavior. These blue dots are trials in which the animal made a movement to lick right. And these red dots are trials in which the animal licked to the left. So you can see this neuron exhibit this robust activity during the delay period that predicted that the animal is going to lick to the right. So now, having recapitulated this activity pattern in an animal model, this now enables us to go in and begin to address long-standing mechanistic questions about how this activity is generated by brain circuit. And my research program is really aimed at understanding how brain-wide circuit is responsible for generating this activity pattern. Our recording experiment found that this preparatory activity is not localized to frontal cortex alone, but rather many connected regions of the brain is also showing similar activity pattern. And this suggests to us that there is a very distributed network that is responsible for orchestrating this activity. And to understand this distributed network, our research program is really motivated by two central questions. So first, we would like to understand which brain region is involved in the motor planning process. And second, we'd like to understand how these involved brain regions interact with each other and what are the individual contributions of specific brain areas. So to get at the first question, to identify the brain regions that's critical for this phenomenon, we would like to take a causal approach in which ideally we could go in and use optogenetic technology to selectively inactivate specific brain regions and do so only during moments when the animal is performing the motor planning. And ideally, and then we can examine the impact on the animal's behavior. And ideally, we like to do this experiment on a large scale by inactivating individual brain regions one by one and systematically survey across many brain areas. However, in practice, this is difficult because current experimental throughput do not really permit this kind of comprehensive survey. So to circumvent this limitation, my lab recently developed a high throughput method in which we could subject the animals to optogenetic testing and in their home cage completely without human supervision. So as shown in this video, this is showing a mouse performing in this system. So you can see in this system, the animal can voluntarily engage and then disengage from behavioral testing in their home cage. And this system runs for 24/7 without any human in the loop. And importantly, the animal is performing the behavior in a head-fixed configuration. And this is very important because it allows us to deliver very precise stimulation to the targeted region of the brain during the behavior. And now by having several dozen of these systems running in parallel, this now gives us a throughput to examine many brain regions in parallel. And now we have the throughput to begin to perform a brain-wide screen of the key regions that might be important for this behavior. So once we identify the regions that's important for this behavior, our next question is to understand the interactions between these regions. To get at this question, my lab developed methods in which we make very targeted perturbations to specific regions of the brain that has been identified. At the same time, we make large-scale recordings using electroarray in other connected brain regions. And this approach allows us to make very targeted perturbations to specific nodes within this distributed network. And at the same time, we can record the network response to these perturbations, from which we can begin to infer the specific functions of each of the network nodes. And over time, this kind of experiment is beginning to reveal a very complex picture of how multiple brain regions interact to mediate behavior. And in the remaining few minutes, I just want to highlight two studies that came out of this work. In the first study, we examined the brain region cerebellum. So the cerebellum is a hindbrain structure classically to be known to be important for controlling ongoing movement as well as body posture. Our study found that surprisingly, the cerebellum not only controls ongoing movement, but it also contributes to the planning of the future movement. In this experiment, we were able to manipulate the cerebellar activity specifically during the delay period when the animal is not physically moving, but they were contemplating their next move. And by doing so, we can examine the cerebellar activity and its impact on frontal cortex. And what we found is that surprisingly, when we transiently silence the activity in the cerebellum, this completely disrupted the motor planning activity in frontal cortex. So this and other experiments in this study together show that the cerebellum has function that extends beyond that of online motor control, but this region also contributes to the planning of future movement, and it does so by reciprocally interacting with the frontal cortex. Now, in parallel, in a more recent set of studies, we have discovered a previously unappreciated novel organization in frontal cortical networks that we think is critical for the robustness of the cortical function. So what do I mean by that? So the robustness is illustrated in this example neuron. So in this example, in this experiment, we were recording the activity of neurons in frontal cortex when the animal is engaged in motor planning. And on some occasions, we provided perturbations to the animal by transiently disrupting their brain activity. And we did so by using optogenetics to transiently silence the activity of hundreds of thousands of neurons within one cortical hemisphere. And what we found was that very remarkably, the frontal cortical network seems to be able to readily tolerate this kind of perturbation. So that is shown in this example neuron. You can see the perturbation here strongly disrupted activity in these neurons. And yet, quickly after the perturbation, the activity rapidly recovered. This shows that the frontal cortical network has built-in mechanisms to readily tolerate perturbations. And so what could be underlying this ability to withstand perturbations? Well, in general, if you want to build a robust system, one way to have robustness is by building redundancies. So if you have a very large network, and if part of the network is being perturbed, if you have other part of the network performing the same function, those intact part of the network could provide a redundancy by taking over the function, and therefore rescuing the perturbed function. And in fact, we found similar principle is at work here. And here, in this case, it's the other cortical hemisphere that is providing the redundancy of function. To illustrate this, what we did is we recorded the activity in the opposite hemisphere. And what we found is that if you perturb activity in one cortical hemisphere, the activity of the other cortical hemisphere is minimally affected. And so the two hemispheres has this organization where each cortical hemisphere is behaving as almost independent modules where it is able to function independently of the other hemisphere. And by having this kind of modular organization, now the intact hemisphere could provide this neural redundancy to rescue the perturbed function. And we think that this similar principle is actually could be underlying the robustness of brain networks to acute insults and possibly chronic neurodegeneration at the early stage of the degeneration. And curiously, we also found that, intriguingly, some mice have a totally different organization in frontal cortical networks. So it turns out that some mice, a subset of the mice, do not exhibit this modular organization in frontal cortical hemispheres. And in these mice, if we provide a perturbation to one side of the brain, the activity in the opposite hemisphere is strongly disrupted. And in turn, the other hemisphere do not seem to have this redundancy. And by failing to do so, these mice were strongly affected by this type of perturbation. So now we're examining the underlying cause for this individual variability, and we're examining the impact of aging and neurodegeneration on the modular organization of brain networks. Okay, just to summarize, my lab has been developing a mouse model to study the neural mechanism of volitional movement. We found there is a distributed brain network that orchestrates preparatory activity, which is responsible for goal-directed volitional movement. We found that the frontal cortex is engaged in a reciprocal interaction with the cerebellum, and this interaction is critical for motor planning. And finally, we found a novel redundant organization and modular organization across brain circuits that we think provides a mechanism that supports robustness of neural function. So I just want to conclude by thanking the people in my lab who did most of this work. I want to thank my collaborators and as well as my funding agency, and particularly the McNear Foundation for providing critical support to my lab when I was first setting up the lab. Thank you.
Awesome, okay. Really fantastic talks. So finally, we have Dr. Sun. Here we go. Okay, timing in resilience. Thank you for all of your resilience.
Thank you. I'll stay on time since I'm talking about timing. We started timing at different scales. On the shortest time scale, we studied circadian rhythm, the 24-hour timing system building in our body. And this is controlled by the circadian clock. This is a diagram of the molecular circadian clock composed of several different transcription factors forming negative feedback loops. All these transcription factors have a very robust oscillation at both RNA level and protein level in almost all the cells in our body. And the central clock, or master clock, is located in hypothalamus, the suprachiasmatic nucleus, SCN, that can be entrained by the light signals through the eye and then synchronized with peripheral clocks through neuronal or hormonal signals. The take-home message for the first part of the story is that we found that disrupted circadian rhythm may underline a down phenomenon, clinical phenomenon observed in diabetes patients, roughly affecting half of the diabetes patients. We measure the blood glucose levels using continuous glucose monitor, and we can separate type 2 diabetes patients into two groups, those with down phenomena and with those without. You can see clearly there's a separation in the early morning and after breakfast glucose levels. And then we took blood at multiple time points, and we look at the circadian clock gene expression, and we found the most robust changes coming from reverb, the circadian clock gene, and their pattern is altered between these two groups. And then we found that this gene is very highly enriched in the SCN in the hypothalamus. So we knockout approach in mouse models, and the SCN is mainly composed of GABAergic neurons. So we knock out those genes in GABAergic neuron specific manner in mice, and we find that they can replicate down phenomena. So ZT stands for that Gabor time. ZT0 is when light is on, ZT12 is when light is off. And those mice shows a very robust glucose intolerance at ZT12 in the evening. That corresponds to, for human, it's the dawn, where at the onset of the activity, waking up. And this does not happen in the other time points. So that replicates this dawn phenomenon in type 2 diabetes patients. And more detailed metabolic tests shows that this is due to the impaired insulin sensitivity, especially at this time point. And this is mainly coming from the failure of the insulin to suppress hepatic glucose production at this point. So there's a connection between the brain and the liver. And that is underlying this phenomenon. To look at this neuronal mechanism, we did EFI study look at neuron firing activity, we found the biggest difference between knockout and wild type is temporal pattern is very consistent with the metabolic phenotype temporal pattern, with the highest difference coming at the evening. And we know this SCN neuron firing activity is important because we can use chemogenetic approach to either specifically activate those neurons, SCN GABA neurons, or suppress those neurons. And we find that if we activate those neurons to mimic what we found in the knockout, we can replicate this phenotype, specifically in those ZT12 time point. And conversely, if we suppress that, we can rescue it in the knockout. So suggesting that this neural activity, the temporal pattern of the neural activity is both sufficient and required for this anticipatory mechanism. And we also show that the reverb, as I said, is a very robust gene expression. It's almost like 30, 50-fold difference between day and night. We knock out a gene, we saw something, we always wonder, well, is this really due to the temporal pattern disruption, or rather you just you disrupt everything, you probably cause something developmental defects. So we rescue it with inducible re-expression of the reverb, but in a phase-specific manner. So if we put those genes back into the phase that they normally express, we can rescue it. But if we put back in a different phase, we cannot. So that really suggests that this temporal pattern of gene expression in the hypothalamus drives the systemic insulin sensitivity. That disruption of that temporal pattern might underlie the down phenomenon in type 2 diabetes. And at a longer time scale, we are looking at how hormonal changes can alter neurocognitive functions. Many hormones bind to nuclear hormone receptors, and those nuclear receptors then bind to co-regulators or co-repressors, which then recruit histone acetyltransferase and deacetylases that modifies epigenome and change gene transcription regulation. So the NCOR has a domain called deacetylase activation domain that activates the enzyme activity of HDAC3. So we studied this complex using a mouse model called NS-DEAD mutant. So this mutant disrupts this interaction and renders HDAC3 enzyme dead. And those mice don't show overall developmental defects, but they have a very robust learning and memory dysfunction. We mapped the circuits. We found that this mainly due to the LH GABA neuron difference, because if we delete in that region, we can replicate the learning deficits. We mapped the circuits from LH GABA neuron to hippocampus C3, and use both in vitro and in vivo approaches. We find this circuit is important for the synaptic plasticity that through it creates a link between hypothalamus and hippocampus. And at the behavioral level, we found that this LH lateral hypothalamus GABA neuron projection to hippocampus C3 is both required and sufficient to cause memory deficits. If we overactivate that, as we found in the NCOR loss of function mice, we can cause memory deficits in wild-type mice. While if we suppress it, we can rescue the memory deficits in the knockout mice. So these studies delineate hypothalamus to hippocampus neural projection that is regulated by nuclear hormone receptor regulated co-regulators that we think maybe provides a epigenomic mechanism that link early life exposure to later life neurocognitive dysfunction because this complex is involved in MACP2, methylated DNA binding protein readers, as well as many ligands that could be environmental toxins or chemicals or drugs or endogenous hormones. So we think this could be a hub that incorporate those environmental changes is through epigenomic regulation and provides a long-term neurocognitive dysregulation. And further evidence suggests that in children with autism spectrum disorders or intellectual disability, there are sporadic mutations in those complexes, so providing further genetic evidence that this complex is important. And finally, on the longest time scale, we study transgenerational, intergenerational gene-environment interaction. We use inorganic arsenic, a commonly found environmental toxin, as a prototype environmental stimulation. And we use exclusive paternal exposure diagram because we want to look at the cross-generational effects earlier rather than later. So we expose mice with inorganic arsenic that is human relevant in contaminated areas in drinking water. We expose them with, we only expose the male mouse at the F0 generation. And then we look at the F1, 2, and 3 generations, in both male and female. All the other mice are unexposed except the F0 male mouse. And in the F1 generation, we see quite robust glucose intolerance and pyruvate intolerance, suggesting they're more susceptible to diabetes. And this effect is mainly due to the changes in the liver, because we can see the liver glucose production is changed in the isolated hepatocytes, suggesting this is cell-autonomous effects, and some gluconeogenic genes are upregulated in the female liver, F1 liver, but not in the male. So this is sex-specific effects in the F1 offspring. And we know this is important because if we can use a drug that blocks gluconeogenesis pathway in the liver, and that can rescue this phenotype in F1 female mice. And in F2 offspring, we saw a growth retardation. And this effect is associated with increased energy expenditure and some morphological changes in the brown adipose tissue that is associated with thermogenesis. More interestingly, in F3 offspring, the phenotype is flipped. Instead of having less body weight and body fat, those at F3 offspring, they have a slightly higher body weight, which is exacerbated in the presence of high-fat diet. So the environment exposure produce sex-specific effects at F1 offspring, and then generation-specific effects at F2 and F3 offspring. So I would like to thank people who in my lab did this job, including Wenjun, Guolian Xin, and Yingyun, as well as collaborators in Baylor and throughout the nation, my former mentor. Our work is supported by core facilities and centers at Baylor. Thank you. Thanks.
This concludes the presentations by the DeBakey Award winners. And I think concludes our feed out to the community. Is that right? Thank you so much for your attention, and I hope you enjoyed this fine work. We can invite all of the awardees up to pick up their medals and their their lovely certificates, which are shown here. Beautiful. So please come get those. And then there is a reception out in the DeBakey Library for those of you who attended, so please join me.